Welcome back to this next video in which we are discussing coronary artery disease. Okay, so in this next video what I want to talk to you about is the biomarkers for myocardial infarction. So one question that might pop into your mind is, how do we tell the difference between someone suffering from unstable angina and someone suffering from a myocardial infarction, either a subendocardial myocardial infarction or a full transmural myocardial infarction? Because remember, the symptoms are going to be very much so the same, okay, because both in unstable angina and in myocardial infarction you're getting ischemia to a portion of myocardium that results in lactic acidosis over there. It's just the severity of the lactic acidosis is going to differ between them. So in unstable angina it's not going to actually end up killing the cardiomyocytes, whereas in myocardial infarction it is going to end up killing the cardiomyocytes. But of course, the formation of this lactic acid and the drop in pH is still going to be activating the pain neurons in unstable angina and myocardial infarction just alike. So you're still going to get the crushing chest pain that refers up to the left shoulder. Okay, so how do we actually tell whether someone is just suffering from unstable angina and their cardiomyocytes have not actually died, the lucky people, from someone who has actually had coagulative necrosis occur in a portion of their cardiac wall? Okay, well this is where we use the biomarkers for myocardial infarction, which can be found in the blood. Okay, so the biomarker that is mainly used now, or the biomarkers that are mainly used now, are the cardiac troponins, specifically troponin I and troponin T. So what I want to explain to you is what these proteins are and why they're going to go up in the blood when you suffer from a myocardial infarction, and how they're not going to go up in the blood when you're just suffering from unstable angina, and therefore how we can actually diagnose someone as having suffered from a myocardial infarction and we know that cardiomyocytes have died in them. Okay, so the topic of biomarkers then. So as I say, to diagnose someone having suffered from a myocardial infarction, what you can do is test their blood and see whether their blood contains these troponins, which are known as troponin T and troponin I, and they're the cardiac specific ones. So firstly, let me explain what these proteins actually are, and these are their abbreviations. So the TN here is just an abbreviation for troponin. So I want to remind you from basic physiology of what these proteins actually are, and then I'll explain why they go up in the blood in the case of myocardial infarction, and you can probably guess, and don't go up in the blood in the case of unstable angina. Okay, right. So what are these proteins then? Well, they are part of the troponin complex. Now, what is the troponin complex? Well, the troponin complex is the complex which attaches onto actin filaments tropomyosin proteins. So let's start by talking about what actin filaments are and work up. So cardiomyocytes have the ability to contract. They have the ability to reduce their length. The reason that they can do this is that they have a huge number of contractile machineries within their cytoplasm. So inside of them they have lots of contractile units known as sarcomeres. And these sarcomeres, one of the key filaments that participates in that is actin filaments. So let me just remind you of the structure of an actin filament. So actin filaments are absolutely key filaments inside cardiomyocytes for the contractile machinery that's actually capable of contracting the cardiomyocyte length down. Okay, so actin filaments then, they are huge, great protein fibres, and they're made up of absolutely loads of actin proteins. So they're polymers, if you like, of actin proteins. So actin itself as a protein is a little globular protein, and to create a great big fibre or a filament, what you have to do is you have to polymerize these together to make a great big chain. And in fact what you do is you make two chains of actin effectively, actin polymers, and thread these around one another. So let me now start to draw an actin filament then. So this is what an actin filament is going to look like. So here is an actin monomer, here's another actin monomer, here's a third actin monomer, and it continues on like so. Okay, and you're also going to have another, so these are lots of actin monomers polymerized together. These blobs, each of them represents an actin protein, and we've polymerized them together here. And there's going to be another one of these chains of actin monomers alongside this one, and they'll intertwine. So here is another one, like so, and now this one's going to go behind, so there'll be another one behind this. So I'll sort of try and draw it like that. So this is an actin monomer behind that one. Okay, and then the other strands popping out back again here, like so. And you can see that the two chains of actin monomers polymerized together are going to intertwine in this way. So this one's now, this chain here, is now going to go behind this other one, like so. So it'll probably be helpful if I colour these in. So here is one of these actin polymers in green. So I'm colouring in all of the monomers in this case in green, like so. So here's one of the actin polymers, like so. Okay, so there's one of them. And then the other one I'll have now in blue. Okay, so again, this is a polymer of actin monomers, like so. And this structure goes on and on. Obviously, I've only drawn a little bit of it. And this polymer of actin monomers, this is known as an actin filament. And these are key components of the cytoskeleton within cardiomyocytes, and they're key parts of the contractile machinery. Okay, so I'll just remind you of the structure of a sarcomere to put this in context. So if we have our cardiomyocyte here, the reason the cardiomyocytes can contract is that their cytoplasm is full of lengths of sarcomeres, and a sarcomere looks like this. So I'll just draw one sarcomere, and I'll draw it a bit out of scale so that I can actually show the detail of it. So here are the two edges of a single sarcomere, and they are made up of proteinaceous discs known as Z discs. Okay, so each of these is representing a Z disc, and you'll have loads of these Z discs within the cardiomyocyte, and suspended in between them will be sarcomeres. Okay, so the Z discs are going to have lots of actin filaments attached onto them, like so. So here are the actin filaments, and I've made those ones a little bit too long potentially for my sarcomere is going to look a little bit wonky, but never mind. So here are my actin filaments attached onto the Z discs, and these have the structure that I've just described to you. Loads and loads of actin monomers all polymerized together into two chains which wrap around one another. So this is an actin filament, and now they're just being represented as blue lines there. And then in between, suspended in between these actin filaments that are coming from either direction. So here we've got actin filaments radiating out from this Z disc towards this one over here, and here we've got actin filaments radiating out from this Z disc towards this one over here. Suspended in between the two, forming the connection, you're going to have myosin filaments, which I'll show here in red, like so, which are again made out of absolutely loads of myosin proteins all joined together. So in red here, these are myosin filaments, and what can actually happen is the myosin filaments can start interacting with the actin filaments, and they can start pulling the actin filaments towards the center of the myosin filaments. So imagine an imaginary line down here which is the center of the myosin filaments, and they will pull these actin filaments in this direction towards the center of the myosin filaments, and they'll pull these actin filaments in the opposite direction towards the center of the myosin filaments. And you can see therefore that they'll pull the two Z discs closer to one another, okay, and therefore contract the sarcomere. And inside of cardiomyocytes, you've got loads of these sarcomeres. You have them in great big chains. So you'll have another sarcomere here, another Z disc here, and on each side of a Z disc you'll have actin filaments. So off this Z disc you'll also have actin filaments going the other direction, like so, and off this Z disc over here you'll have actin filaments and then myosin filaments in between the two, forming a great big chain of sarcomeres inside the cardiomyocyte. And you'll have loads of these great big chains of sarcomeres along the entire length of the cardiomyocyte, and therefore when they all contract, when the cardiomyocyte receives the electrical signal and converts that into a calcium signal, you will get contraction of the entire length of the cardiomyocyte. So there's a brief explanation as to how cardiomyocytes contract using these sarcomeres, and a key component of this then is these actin filaments. Now, the reason that the sarcomeres only contract when an electrical signal has actually reached the cardiomyocyte is that when an electrical signal reaches the cardiomyocyte, it converts it into a calcium signal. It allows calcium level inside of the cytoplasm of the cardiomyocyte to go up hugely. And I've drawn this incorrectly here because I was about to draw an arrow of calcium coming in from the extracellular fluid into the cytoplasm, but of course those of you who know anything about excitation, contraction, coupling in cardiomyocytes will know that the main source of the calcium that causes the calcium signal inside the cytoplasm is actually from the sarcoplasmic reticulum, so intracellular stores. So, when an electrical signal arrives on the cardiomyocyte cell membrane, it converts that signal in a process known as excitation, contraction, coupling into a calcium signal. Loads of calcium ions are released from the lumen of an organelle known as the sarcoplasmic reticulum, which is just the equivalent of the endoplasmic reticulum in these cardiomyocytes. And that calcium is then what's going to activate all of the sarcomeres. And the reason it activates them to begin contracting, it has completely to do with troponin. So now back to troponin. So we need to explore the reason that usually sarcomeres are not contracting, and the reason that they're usually not contracting is the myosin filaments are not allowed to interact with the actin filaments. And remember, in order to contract a sarcomere, the myosin filaments have to interact with the actin filaments and actually pull them in a certain direction. Okay? Normally they're not going to be able to do that. There's going to be proteins obstructing the interaction between myosin filaments and actin filaments. Okay, so the next player that I need to introduce you to is the protein tropomyosin. So tropomyosin is a protein made up of two separate polypeptides, an alpha chain and a beta chain, which intertwine with each other like so. So I'll draw them like so. So here are these two polypeptides, an alpha chain, and I'll colour them in separate colours. So one of them here in pink, that can represent the alpha chain, and then another polypeptide, okay, and of course a polypeptide is just a polymer of amino acids, is there in green. So tropomyosin, the protein, is made up of two separate polypeptides, the alpha chain and the beta chain, okay, and it looks kind of like this. And what's going to happen is you're going to get loads of tropomyosin proteins in the cardiac muscle cell here, and you're going to put them over the actin filaments. Okay, so one tropomyosin protein can cover up 7 actin filaments, 7 actin monomers in an actin filament. So if you like, you can imagine putting it on here, and if I continue the picture on so that we've got 2 more actin filaments there, you can imagine putting your tropomyosin protein on here. Okay, and I'll just colour those in. So here are the 7 actin monomers in the actin filament, 1, 2, 3, 4, 5, 6, 7. Okay, and here, which I'll colour in red, this is the tropomyosin protein, and the tropomyosin protein is going to bind over 7 actin monomers in an actin filament, and it's going to block them from being able to interact with myosin filaments. So these 7 actin monomers within the actin filament now cannot interact with myosin monomers within the myosin filament. And by the way, the reason, or the way in which the myosin filaments can interact with the actin filaments is that the myosin monomers of the myosin monomers of the myosin filament interact with the actin monomers of the actin filament. Okay, so by blocking the actin monomers, we are blocking the myosin monomers from being able to interact with the actin monomers, and thereby blocking the myosin filament from being able to interact with the actin filament. And you'll put these tropomyosin proteins over all of the actin monomers. You'll have loads of them, and you'll gradually cover your entire actin filament with these tropomyosin proteins. So you can imagine sticking them on all over the actin filaments, all over the cell. Okay, now how do you actually stick them on? Well, here is the answer. Troponin, here's where troponin comes in. Troponin is the way in which tropomyosin is stuck onto the actin filaments. And sticking the tropomyosin onto the actin filaments is what usually prevents the sarcomeres from actually contracting. So that's the reason that when you've got a cardiomyocyte in a resting state that's not contracting, it's the reason it's not contracting because the tropomyosin filaments are obstructing the tropomyosin proteins are obstructing the actin monomers of the actin filament and preventing the myosin filament from being able to interact with the actin filament. Okay, so coming back to troponin then, troponin is the complex which binds tropomyosin to the actin monomers of the actin filament. Okay, so if you like, it looks almost like this. So if this is our tropomyosin protein here, then what you're going to have is this little complex of 3 separate proteins known as the troponin complex. One that's quite large, which is troponin T, okay, one that's in the middle called troponin C, and then the other one here is troponin I. So these 3 proteins, and I'll just firstly show you tropomyosin. So here in red, this is representing the tropomyosin protein, so that dimer of these 2 polypeptides here, okay. Then these 3 boxes here, these are representing the troponin complex of 3 proteins, and overall this complex is what attaches the tropomyosin protein to the actin filament. Okay, and now the troponin complex is made up of 3 proteins. The one that attaches to the tropomyosin, as shown in this picture, is called troponin T, and for short troponin is just abbreviated to TN. So TNT here is troponin T. The one in the middle, which is the one that actually senses calcium and is going to respond to calcium in order to do something about the tropomyosin when you actually get a calcium signal, which I'll explain in just a moment, that's called troponin C. So here is troponin C. And then finally, this one down here, bound to troponin C, that's called troponin I. And troponin I is then the one which will actually be bound to actin monomers of the actin filament. Okay, so this is troponin I. So troponin I's job is binding to the actual actin monomers of the actin filament. So then you'll have the rest of the actin monomers here that are going to be covered up by this tropomyosin protein. Right, so the troponin complex then is involved in binding these tropomyosin proteins over actin monomers in actin filaments. Now, when a calcium signal arrives in the cardiomyocyte, calcium will bind to troponin C. The troponin complex will change conformation and move the tropomyosin protein so that it no longer actually obstructs the important binding sites on the actin monomers, which the myosin monomers of the myosin filaments actually binds to. So even though the tropomyosin will still overall be bound to the actin filaments, it's no longer actually covering up the essential binding sites of the actin monomers, and therefore the myosin filament gains the ability to actually interact with the actin filaments, and contraction will actually begin. So the sarcomeres will all contract down, and when all the sarcomeres start contracting down, that will contract the overall cell. Okay, so that's a summary of the role then of troponin in the cardiac physiology. Now let's actually talk about its use as a biomarker. So troponin proteins, they're also really important in skeletal muscle cells. However, the actual protein that is used in the troponin complexes in cardiomyocytes is different from the troponin proteins that are used in skeletal muscle cells. So skeletal muscle cells have this exact same system. This is the reason that the sarcomeres inside skeletal muscle cells are not usually contracting, that you have troponin complexes made up of these 3 types of troponins, troponin T, troponin C, troponin I, that are binding tropomyosin proteins over the actin monomers of the actin filaments. And then when calcium goes up in the skeletal muscle cell, the calcium will bind to troponin C and change the conformation of the troponin complex so that the tropomyosin protein is moved so that it no longer obstructs the important binding sites on the actin monomers, and the myosin filament monomers can now actually start interacting with the actin monomers in the actin filament. Okay? However, although for simplicity we just talk about troponin T, troponin C and troponin I, there are different versions of troponin T, troponin C and troponin I. So there are different genes in the human genome that make proteins that perform an equivalent function. So there are different genes for troponin T proteins, different genes for troponin C proteins, different genes for troponin I proteins, and these different genes will make slightly different proteins with different sequences of amino acids. However, the proteins are similar enough that they perform the same role physiologically, okay, which is why we just abbreviate them all down and just call them all troponin T proteins, but the actual structure of those proteins differs. So in cardiac muscle cells, there is a specific type of troponin T, a specific type of troponin C, and a specific type of troponin I used in those cardiac muscle cells that is different from the specific type used in skeletal muscle cells. Even though the proteins perform the identical role, their actual amino acid primary structure is going to be different because they're coded for by different genes. So this is what we mean when we say cardiac-specific troponin T, or cardiac-specific troponin C, or cardiac-specific troponin I. We mean the specific protein that is actually used and performs that function in cardiac muscle cells. Okay, the specific version of it. Okay, so we're talking about an actual specific protein here, rather than a whole bunch of proteins that perform the same function. So when you say troponin T, that is not an individual protein, basically. Okay, if you just say troponin T, that means a protein that performs this function. There's a whole bunch of them. Okay, if you just tell me to write you out the primary amino acid structure of a troponin T protein, I can't do it, because there are multiple different troponin T proteins that have different primary amino acid structures. Okay, so that's a really important concept to understand, that in cardiac muscle cells there is a specific troponin T, a specific troponin C, and a specific troponin I, okay, which is identifiable because of its primary amino acid structure. Right, so now when the cardiomyocytes die in a myocardial infarction, the troponin proteins, troponin T and troponin I, end up going out into the extracellular fluid, and from there they can get into the lymphatic drainage of that area of the heart, or into the capillaries in that area of the heart. Okay, and from there they can of course then end up in the circulation. They can end up in the systemic circulation. So when we then take a blood sample, we can look for the presence of cardiac-specific troponin T and cardiac-specific troponin I, and those two are the specific ones used. So when we're trying to diagnose someone as having suffered from a heart attack, we take a blood sample and we look for cardiac-specific troponin T and cardiac-specific troponin I, and this shows us that there must have had to be myocardial cells dying. It must have been a myocardial infarction, because if it was just unstable angina and you didn't actually have any cardiomyocytes dying, then these proteins would not have been released from the cells into the extracellular fluid, and they wouldn't have ended up in the systemic circulation. Okay, so by looking for proteins that usually are not in the blood and which are inside cardiac muscle cells and not inside of other cells in the body, then we can determine whether cardiac muscle cells have died. So these are the main biomarkers used to diagnose someone with having suffered from a myocardial infarction. We look for blood troponin T levels and blood troponin I levels. Okay, so I hope you understand that. We will have a break here, and in the next video I will take on the incredibly difficult task of trying to explain to you why it is that subendocardial myocardial infarction and transmural myocardial infarction look different on an ECG, why subendocardial myocardial infarction appears as the non-ST elevation myocardial infarction on ECG, where the ST segment of the ECG has not actually elevated, and why when you have a transmural infarction, the ECG shows you an ST elevation myocardial infarction where the ST segment is elevated. Okay, so we'll do that in the next video.